exactly what you need to know. You're learning the instructor. And I was thinking, when you go through slides about the cell membrane, people nod a lot, but then you test them and they don't really understand it. But it's not as complicated as it seems when you just sit down and talk about it. So I was thinking, man, it'd be better just to use a whiteboard. Because how did y'all feel about yesterday talking about, y'all understand the lipid membrane, I mean, I know some of y'all probably have more of a background in that. But the reason we're studying it is to basically see how you maintain compartments inside of cells, because the chemical composition inside a cell is much different than what's outside. But we need that to occur. Because when you look at ions and molecules going through fluid compartments in the body, like interstitial fluids, they pretty much can freely flow everywhere, right? Take a drug, it gets everywhere. But not every drug will get into every cell or every area, right? Because there's membranes that separate that. And you look at things like the blood-brain barrier, that's a really selective membrane. It doesn't allow a lot of things like antibiotics to get into cerebrospinal fluid. That's why they're important. So when we talk about, and people understand that when you say it, but when you talk about the structure, and that's why I was going to, and I'm going to do it, even though it takes time from yesterday's stuff. I'm going to raise this for a second. Because when you just look at the cell membrane, you see these definitions, you say, well, it's a phospholipid bilayer. You start going, well, I need to understand that, and you're trying to figure that out. Well, we can draw it. You can understand it in 2 minutes versus 2 hours. What do you mean when you say a fluid mosaic model? You know? And we're not getting everything on this. That's another problem. So sorry, I'm going to use this side. I'll use that side next time. Yes?
Can you use this side because we have the camera?
Oh, yeah. All right. Y'all need to get a
Y'all
camera.
can rewatch it.
Well played, young legend.
That's pretty good.
That's my assist.
All right. And it's not going to be fancy. I'm not the world's greatest drawer. But if you were in my office, what I would do is go, oh, you don't understand the bilayer? You would say, okay, well, where's it at? The whole board?
It's got the whole board.
So if you take and try and draw it big here, and it works. And you got a cell. And then you got this plasma membrane and the plasmolema around it. Right. So everywhere out here we have interstitial fluid, right, surrounding that cell. We're talking, some of this is just going to be generalities. But when you say the ionic composition here is different, I'll go ahead and just note it. Let's say, so sodium, let's just use one molecule. Sodium, or 2 molecules. Sodium's higher outside, potassium's higher inside. And we're about to get into that. We'll get into that later. But we're maintaining differences between the inside and out. What's that called from yesterday? Yeah, well, yeah, concentration differences, though, is what I'm looking for. So there's a difference, and the difference why that exists is because of this membrane, plasma membrane. So when we say it's a lipid bilayer, it means that we take, y'all remember what the lipid bilayer is made up of?
Cholesterol.
Phospholipids, glycolipids, cholesterol. And I'm not going to draw all of those. I'm just going to draw a couple of, these are the phospholipids. I know that's really small, but marker doesn't work great. Let's do a couple more. Those are supposed to be fatty acid tails. Now, there's a lot you could go into if you really wanted to study this stuff. Not that I'm all into that, but it helps to explain. So that's the phospholipid bilayer. And let's just put, those are glycolipids and cholesterol. We'll put GC, so we know they're there. Well, cholesterol mediates fluidity due to temperature changes. Glycolipids fill in the rest. And so this lipid membrane can actually move. Just like the fluid moves around it inside and out, the membrane itself is kind of fluid. And so that's why they call it a sea of phospholipids kind of moving. And if you were to put a hole in it, they would fill that in very quickly, those lipids. So you have this head of this phospholipid that likes water, and you have these tails that don't like water. And so I hate biochemistry. I mean, I don't hate the person and the teacher and the class. I hate learning it myself. But there's molecular interactions that go on that cause these to orientate that way. Mainly that they're polar and non-polar, right? The heads and the tails. And so they're going to line up that way, then these other two will help this membrane. And so you'd say, well, that's a phospholipid bilayer. But we also call this a plasma membrane. Because when you say plasma membrane, that now includes the proteins. And I once read this, and it's a little too much knowledge, not too much knowledge, is when they were studying these originally, I say they, because I wasn't there and I didn't read every bit of it. But they were talking about it and saying they didn't understand how they found proteins in this membrane, but it's lipids at the same time. So they were coming up with a way to describe that. And they said, well, there's a phospholipid bilayer, but there are, I'll do a squiggly one, there are proteins, remember there's proteins outside, peripheral and integral proteins we talked about yesterday. The cell is going to put those into the membrane. And let's draw the famous, draw like a heart. That would be like a sodium-potassium pump. Sodium and potassium in and out. So the cell can place these proteins in the membrane. And there's a lot of them. Books only show one or two. We're talking lots of different protein or carriers. And so each one of these can be like a proton or sodium-potassium pump, or you could just, which is also an integral protein. Does that make sense? It's, you name, as we study further, you'll see you name voltage-gated sodium channels, leakage channels, all these things we're going to talk about, but they're still just integral proteins. Proteins that the cell has integrated into the membrane. That's the way I would say it in my mind. And so they all exist, but since this is kind of floating, they also call it what they came up with to describe it was fluid mosaic model. It's fluid because of the lipids, but you have all these proteins that are scattered all around the outside they could see when they looked at these microscopes, and it makes it look like a mosaic, like the little chipped glasses and stuff. And so it gives it this mosaic look. So that's why it's called a fluid mosaic model. Just their way of describing it. And so all of that's key to understanding is we're about to talk about movement, how we move things. What if we need sodium to come in? How can that happen? Right? Because sodium can't just cross the membrane. It must have a type. There's different types of these proteins. It must have, I don't want to say carrier, it must have a way to get across. That can be like, we're going to see there are leakage channels, there are voltage-gated channels, there's ligand-gated channels. Remember that 2 pages where it had the 3 pictures on each one? All those different channels will be here to allow that sodium. So it has many ways to get in there, just like all molecules do. Does that kind of make sense? That's yesterday in a recap, really. That's what we're looking at, because once you understand that, then you come. That's probably why the markers don't work, because I leave the top stuff. I don't write on that board often, because it screws the other side of the room, but especially the front row. But you'll be all right. Okay.
I forgot that was cool.
As long as this side's happy, we're good. We're good. Does that clear anything? Does that help at all from yesterday? You don't have to not do it. I'm just saying. I think it does in my mind. Because if not, if you don't understand that part, then rolling into movement doesn't make as much sense. Because now we know the structure of the cell plasma membrane, and we know why things aren't getting through because of those lipids. And I didn't get into charges yet, right? There are charges that make it different. All right, so we want to change let's think of it as we want to change the chemical composition of the inside of a cell, whether that's right now we're talking generalized cells, but it could be a nervous cell, which conduct action potentials, which are electrical potential in the body, which is very different than a normal cell. And muscle cells, skin cells, all these different cells are going to use these processes to change the chemical composition inside of that cell. The cell is going to decide what it wants to do. Remember, it's going to collect that stuff from the extracellular fluid, interstitial fluid. So many ways we can do this. We can do passive processes, or we can do active. ATP. ATP is the body's energy. So any passive process will not use energy. Putting sugar, the reason I made an analogy and I didn't do it well yesterday, is when you put sugar into tea, that's simple diffusion. No energy is required because a concentration gradient exists and there's nothing to separate. You know, if you were to separate the sugar in the water with a membrane, it would have to be a membrane that allows water or sugar to go through it. You know what I mean? You could have, it's not permeable to either one of those, they wouldn't simply diffuse. You'd have to use energy to do it. Right? That's why heating it up is a form of energy. If you heat it up, you can increase the amount of diffusion. But in a passive process, there's no energy, and that includes diffusion. We'll talk about osmosis, facilitated diffusion, and then what's called filtration. We don't spend as much time on filtration. Filtration is coffee. We'll see that down in the renal system. You got to filter things through. You've already started learning about capillary, maybe. I don't know about pressures. No. You're going to learn that there are proteins in interstitial fluid, and there's proteins in intravascular fluid, and it helps with the exchange because of osmotic force. That's not my class. Then you have active processes where you find things like primary active transport, secondary active transport, and then bulk transport. And we'll talk about all of these. So the first one up is diffusion. So passive process that moves particles right, into solution, basically. So we're taking things from an area of high concentration to lower concentration. And we already mentioned the T. Well, we can increase the amount. It's called the steepness of the gradient, but depending on how much we have of each, whether it be water or some molecule, how much they will diffuse and how fast it occurs is by how steep. What's steep meaning the difference? How big a difference is it? Right? And then we can increase the temperature. What's the surface area? How far does it have to travel? And of course, that surface area and diffusion distance, pretty much the same thing. Right? And so then you get your beaker where somebody dropped blue dye in it. Eventually, it will reach equilibrium. Right? As long as there's not more than one of the other. All right, so diffusion is easy to understand. But under diffusion, also, we have osmosis. And osmosis is similar to diffusion except for one thing. It only involves the movement of water. Diffusion can be molecules of water as it's going, but if you're just talking moving water through a semi-permeable membrane, that's osmosis. When you say semi-permeable, that means that the only thing that can get through that membrane, well, I don't like to talk in absolutes, but we can sometimes when we're learning it. Osmosis has a semi-permeable membrane that only allows water to go through it. Any molecules that are in that water won't go through that membrane. That's what we're talking about. So it still follows the rules of diffusion. You must have a concentration difference, gradient difference, and we're always going to move from a compartment of a higher water concentration to a lower solute concentration. Or sorry, you have a higher water concentration but less solutes, and it's going to move to an area of concentration where the solute concentration is lower and the water concentration is higher. I'm going to show you that in the beaker. So here are your requirements. Semi-permeable membrane, at least one osmotically active solute that is on the higher concentration, can't pass through the membrane, and then osmotic pressure. So what does it mean by osmotically active solute? Okay. We're not biochemistry, but we still have to understand that. It is molecules that are osmotically active, like glucose. Think of them as, you know, they can take, they're gonna suck water to it. It's like it's collected. So glucose is going to attract water to it. It has a pull. And so a great example of, I always think of osmotic forces, is something simple, like cat pees on your carpet. What do you do in your, I don't have carpet in my house anymore, so ripped all that out. I tell y'all things that y'all don't need to know. That's fun. Well, what do you do like cat pees on cardboard? What's one of the things you can do, especially you're running out the door, you know that water spot gate's gonna be backed up again, and you're running, but you can't clean it up right then. Even if you are home, you're gonna dump salt on it. You don't know that helps to soak it up. So you take your big thing of salt and dump it all over that, and what's gonna happen to the salt? It's gonna turn yellow because it's gonna soak, we call it soak, it's gonna soak all that urine up into the salt, right? Salt has an osmotic force that's doing that. If not, it would just lay there on top of it. No different, don't make it complicated, it's no different than in the body, is different molecules have different osmotic forces. Like you would say glucose is highly osmotic, sodium's osmotic, but glucose is really osmotic. Right? You don't believe that, go eat like 3 or 4 bags of sweets and start seeing what your poop looks like. You're gonna start bringing a lot of water into your bowels and stuff. Or you think about diabetes, they pee a lot, right? Too much glucose, which should never leave. You shouldn't have glucose in your urine, but because in diabetes you have too high of glucose, you're spilling water out with it because glucose is going to take water with it. I think I spent too much time on that, but it helps. And because glucose is osmotic, that creates a pressure. It's pressure driven. Osmotic, that's where the osmotic pressure line comes in. So normally I don't say all that in that slide, but it helps to understand. And so if you took like this U-shaped beaker as an example, and we had water molecules on the left side here, and then we had water and solute molecules on the right side, and we put a semi-permeable membrane in the middle, and that was our starting condition. What's going to happen, right? Let's say, you got to picture this, that we had a glass of just water, then we had a glass, and I'm going to call those yellow, we'll call them sodium. We could use glucose, right? We'll call them sodium. And so you have a glass full of water, you have a glass with water and some sodium in it, and you have a U-shaped beaker with a semi-permeable membrane. And picture if you dumped them in there at the same time, initially they would go to what that first picture looks like, right? But over time, what's going to happen is all of these molecules are osmotically active. If they are, which sodium, glucose, they're active. So there's molecules on this side. So that's going to pull the water. Y'all just got to yell at me when I write on that screen, because it's going to mess up every time. It's going to pull the water this way, through the membrane. But remember, what does the concentration difference do? Remember, we use concentration gradients. And so, just so you're thinking right, if the membrane wasn't there, what would sodium do in this case? The yellow molecule. They would just equalize. The membrane's keeping them from doing that, but because those molecules are also osmotically active molecules, they're pulling water through. So we're pulling more water into the right side, and now you're going to see that level rise. Until you don't have any more osmotic pressure. It can't pull anymore, right? You can only soak up so much pee with the salt you put on there. They have to wipe it up, vacuum it up, and do another. Alright. So that's osmosis. But what if we wanted, remember that membrane is permeable to water, just not solute. So what if we put a cap on this one, we could actually measure, still doing it, we could measure that pressure that's being, saying that because I messed up the drawing. If I cap this and there's osmotic pull through here, pushing up, I could measure that pressure, right? If I had the right equipment. That's osmotic pressure. We could see how much it's pushing up on that cap. In other words, how much it's pulling that way. What if I took and forced this water and molecules back this way? I could push water back through that membrane, but not the solutes, right? Because solutes can't get through anyway. So I can push the water back, and what do we call that? We drink it every day. Reverse osmosis. Right? I'm one of the few people left in the world that still drink out of a garden hose. I know y'all don't. So actually, that is a complete lie as of 2016. Up until then, I did. Outside, anybody drink out of the hose still? It's good for you, a little bit at a time. But I actually had a kidney transplant in 2016, so I'm not that stupid. I don't do that anymore. It's kind of killed my water skiing days, but I don't like to get in warm lakes anymore either. Lost the train of thought now.
Warm lakes.
Warm lakes, reverse osmosis. Oh, we all drink this filtered water now. We use reverse osmosis, force it back through. Makes the water cleaner, I guess. It gets rid of stuff, but tastes better. Alright. Drinking out of the hose, for those of you who don't know, they're like, who would drink out of a hose or get so sick? Here's my analogy. You know, I like analogies, and I go home every day driving going, why do I say that stuff to them? They don't care. They want me to talk about anatomy. But I like to give you life skills too, even though some of y'all are as old as me, bro. Maybe not. I'm not taking a poll to see, so I don't want to know that. No, I don't care all day. We're all dying. I mean, you know what they say. Eat right, exercise daily, work hard, still die. Now I lost what I was thinking again. Right, what was I saying? Oh, the hose. You go to Mexico today. You go down, you go outside of the limits and against regulations, and you cross over down at South Padre, walk across that border. Don't do that until you get out of here. It's not safe right now. You cross that border, it's safe enough. It's Mexico. Mexico's not that dangerous. You go into Mexico. Are you going to go into Mexico today and start drinking water right out of, or even in the restaurants? Probably not a great idea. You're not used to the bacteria and stuff that are there. We're military. We go to a lot of different countries. But if you go to Mexico today and start eating the food, being in the environment, eventually you're not going to have those GI problems and you will be able to drink the water. So my analogy is drinking the hose, yeah, you may get a little bacteria or something with it, but building up that immune system, you're exposing it to it. So that's how I defend drinking out of the hose. I actually have a bottle of water that says hose water from talking about that from a previous class before the transplant. Okay. Let's get back on track. Sorry. So we did reverse osmosis. Now tonicity is looking at, well, that's great that these things are moving, but what about how they're moving into and out of cells, and what does that cause? Tonicity is, by definition, if you read, a measure of a solution's ability to change the cell's water content by osmosis. You're going to cause osmosis. In other words, we're going to cause water to move into or out of a cell, into or out of a body compartment, intravascular, interstitial, that kind of stuff. The movement of the water. We're just looking specifically. And so the function of this relies upon a concentration of osmotically active solutes. Water is not going to move unless there's some type of hydrostatic, like blood pressure pushing, or there's an osmotically active solute that's bringing it towards it. And so we describe this looking at different fluids. So the relative tonicity of intracellular fluid versus the type of fluid you might put there. Is it isotonic? Is it hypotonic? Is it hypertonic? So here's another example where you say, well, here's 3 beakers. Each of them have 1 cell in it. And into that beaker, we're going to look at the first one. It's already in there, but I like to imagine we're going to pour. We're going to have 1 cell in a beaker, and if we pour an isotonic solution in there, what's going to happen is that blood cell, we're looking at the movement of water into and out of the cell. there will be no movement of water really into or out of the cell. Say that another way, let's put 5, since we know sodium is osmotic, let's put 5 sodiums in the water that we poured in. And there's 5 in here. There's no, forget about the cell membranes and all that right now. I didn't get the N in there, sorry. 5A plus. There's 5 sodium inside, there's 5 sodium outside in the solution that we poured in of water and sodium. They're equal. So in that case, even if that cell membrane has, is semi-permeable, there'll be no movement because there's no difference in the concentration gradient. Make sense? That's IVs. Before 0 day aerosol, you're up in the barracks with medics giving each other IVs. Hopefully you're using isotonic solution. In your combat medic, they still have combat lifesaver bags in like all the vehicles. I've been out a while.
Should.
Should. I showed up in Afghanistan.
They've been wrapped up.
Yeah. I showed up in Afghanistan at Bob Condu's, Bob Deadwood, what it was originally called. That was the better name off the show, Deadwood HBO special. It was in the middle of nowhere. And when I got there, we only had a couple of trucks. There's only 16 of us, and we're transitioning right seat, left seat with this group. And I go out, and I look in the vehicles, and not only were they leaving them in there while it's freezing outside, terrible idea to buy IV bags, but they had hypertonic solution in them. Not all, but some of them in there, somehow they had gotten hypertonic solution in there. That's a terrible, can kill you idea. And that's why I mentioned this. So now when y'all go, you'll check too. Because you'll be amazed what winds up in combat lifesaver bags. It should or shouldn't be in there. Or even medics aid bags sometimes. I'm not putting down medics. I was a medic for 10 years. So I'm saying, we didn't know what we didn't know. Is that fair enough? Right, now we know. Let's look at hypotonic. We'll get to hypertonic. So if I have a hypotonic solution, that means that the solution I pour into the beaker has, I keep forgetting the N, I just don't want to get sodium in. 3 sodiums, and let's say the cell still has 5. In that case, which way is the water going to move? It's going to go into that cell. And if that continues, you could have that red blood cell, in this case, could go through hemolysis, meaning it could burst. Right? If there were so many osmotically active solutes inside of that cell, and you put a hypotonic solution there, the cell is a much higher osmotic pull. You can physically cause it to pull in there and burst. I don't know why I'm getting into so many examples today. I'll give you an example. This time from Kosovo. We were leaving Kosovo, and they said, hey, we're going to make everybody redo their PPD test, or ghost test. So I got with the medics over there when I was there. I said, listen, we're going to do these really quick. So does everybody know how to do a little wheel? And some of them said yes, some said no. And they didn't know me, but I'm just asking for reference. We're going to practice anyway, right? And I went and grabbed what I thought was normal saline, but it was actually sterile water. And we're sitting there, and they're doing it on each other, and I'm watching. And they're bitching. Sorry, can you use that word here? They're going, man, that hurts. I'm like, it's a little will. What are you talking about? It hurts. And they're like, it's killing me. They said they could feel it all up and down their arm. And I'm like, you must be doing it wrong. Anyway, eventually enough of them complained. I said, well, just do it on me. I don't believe you. We've done these a million times. And they did it on me. And I'm trying not to show it, but I'm like, that hurts. That hurts. And what was happening is that sterile water being lower was causing cells to burst because of this hypertonic stuff. It was causing those cells to burst right underneath the skin. So, I mean, it goes way up to about 5 minutes. But it's kind of, you can do that to your medics one day. It's not going to hurt them. Don't delete that off the recording. Might get me later. That's okay. One of the guys before Afghanistan. Somehow the embedded training team before us, they went out to teach some of our gunners how to shoot down in this wadi. And for some reason, they had an AT-4 that was unaccounted for. And they decided before they leave and while they were out there, they'd shoot it off. That didn't go well. That was, we were in the middle of nowhere, but that was noticed. The Germans are up there with you too, monitoring things. They noticed. And guess who they report that to? The head command. They don't call you. Anyway, that dude got in all sorts of trouble. Where did you get it? Yeah. We heard an explosion. Okay. I gotta stay on track. Isotonic, you're not gonna have a difference. Hypotonic, water's gonna move into the cell. That's normally a good thing. We can use that. Hypertonic, in that case, that's where we reverse it. I'm just gonna put a 5 for time's sake. We have 5, we have 3. In that case, the osmotically active solutes, you just pour it in with that solution or higher, and so the water is going to move out of the cell, and it will go through crenation or shrink. Yes?
Second row inside. Yeah, I have a question. Taylor.
Me?
Yeah.
Oh, thank you. So I've always struggled with this concept. I can never get it right. Is it because?
Well, that doesn't bode well for the next minute.
Yeah, we'll see.
Sorry, y'all get offended when I do that to you. You gotta let me know. 'Cause I just like to lighten the air. No.
Bring it.
Let's hear it.
It doesn't make sense to me because it feels like it's not, the sodium's gonna go from a higher concentration to a lower concentration, and so every time I think of it having more sodium in the cell than outside the cell, it's going to diffuse. But in this case, the water's going into the cell.
Water follows salt.
It does. So we're not talking about the sodium. We're just talking about the water following the salt, and because the water's following the salt, then it lyses.
Yes. But what we're partly just, it's a good question, but the part you're leaving out is what I don't keep saying, is we just talked about osmosis. We're still talking about the movement of water, and so picture that the semi-permeable membrane is surrounding this. And so in that case, only water can move.
Okay.
So sodium's gonna stay out here. So because sodium's higher out there, and like he said, it's osmotic, it's gonna pull water out of the cell instead of letting it equalize.
And so with hypertonic, it's getting pulled out of the cell, and with hypotonic, it's being pushed into the cell.
Yeah, because I dumped a highly salty water onto that cell, and the cell doesn't have as much sodium in it. Because sodium's so osmotic, it's going to take, even though there could be more water outside of the cell than in, but if sodium's higher outside, it will still pull water out of that cell. That makes sense? No, we're not leaving till you got it, because it's been years. You haven't got this.
I know it's such a silly thing, but it's always been something I can't wrap my head around. Just because of the numbers thing, like I think I'm thinking of the sodiums moving instead of the water moving and following the sodiums. So.
It's not gonna move. It's like having a block of salt sitting outside that cell. It's just gonna pull. Sodium's gonna start, and it's osmotic. It's gonna start pulling on that water to all the cat feed.
So if you were to drink a large amount of distilled water without all the minerals and sodium in there, would that cause a hypotonic solution?
You dilute out your body fluid compartments. Yeah, you don't want to do that.
No, for sure, totally agree. I just wasn't sure, so it would cause a hypotonic solution because there's no sodium from that water you're ingesting, and so that water will just continue to pile in the cells.
Yeah,
Am I understanding that right?
and intravascular, you're going to be hyponatremic, and sodium is a very narrow range in the body, and you're going to be in bad shape really quick. I'm trying to generalize, but if you ever worked inpatient medicine, sodium levels are highly regulated. Like if you have a patient who's hyponatremic, hypernatremic, we got to monitor their water intake, but distilled water wouldn't do us any good. Works great in your iron though. Sorry, that's the only thing I think of for distilled. Yes?
So when we're looking at the semi-permeable membrane, are we looking at a quick action homeostasis versus like a long-term homeostasis? So water can shift quickly
Yeah, if we could narrow, I don't know a time frame, but did y'all hear that? Talking about how fast water moves compared to how fast molecules can move. And that's exactly right. A molecule like water can move through that membrane back and forth very easily, but things like sodium need channels, leakage channels, it needs a channel to open to move. So it's not going to move near as, I don't want to say near as fast, as easily. Because we can move molecules fast. But we're going to need, it's a great question because it leads us back to, in order to move molecules really, really fast, the main thing we're going to have to do is use energy, ATP, in a sense. Right now?
I'm just looking at it as a similar, like, compensatory mechanism as with balancing the hydrogen atoms, is your respiratory tract is more of your immediate response, and then your endocrine cycle works slower but more long term.
Yeah.
It's a similar dynamic for homeostasis.
Yes, but we can also shift, if this makes sense, body fluid compartments fairly quickly. You know, let me give you an example. I mean, it's relative to the time, but let me give you an example of this if it helps us. Like, why in the world would I want, I'm in Afghanistan and a bomb just went off. I'm not going to do this necessarily out in the field. Why would I ever want to use a hypertonic? Why would I always want to isolate? He wants to add into this. Yeah, intracranial pressures, you can pull water out of there, especially. What if, forget blast injuries, let's say your blood pressure is bottoming out because you're bleeding out. You could use, and you want to use like an isotonic solution so you can add fluid to the intravascular volume in your vessels. If you don't have fluid in your, we're getting way into physiology, but if you don't have fluid in your vessels, you can't perfuse your organs. But as a last ditch, I'm going to call it last ditch. Yeah, what's that? Yeah, head of starch, or we can argue that all day. But, I can give you, I'm trying to keep it on the Barney level. If I want to, let's say I don't have a lot of it, isotonic solution, or you're cratering quickly, does it make sense that I can give you a hypertonic solution? And where am I going to rob the water from? The cells. So at the expense of the cells, I'm going to increase your intravascular volume. That's why you brought us right back around to. That's why having hypertonic solutions in your CLS bag is not a good idea. Because even I don't know that I'm comfortable doing that. The only reason I would do that is if I have no other option to get someone's intravascular volume up, I would use it. Other than that, and I know I'm not getting sued on the battlefield. So. Okay, did we get close to your answer for your question? I strayed it off a little bit. Anyway, it's interesting stuff. That's funny. That's the first time in a class I think that we made phonicity actually interesting. It's just the movement of water. I thought it was interesting. I like talking, so at least in here. Once I leave here. Alright. So what we are supposed to be talking about though is movement of molecules across the membrane. And I'm bringing everybody back. And we were talking about diffusion, passive diffusion, osmosis. I didn't put a slide for filtration because we kind of understand that. You have a membrane that filters things through and there's pressures involved. But there is one more called facilitated diffusion. Okay, facilitated diffusion is a little bit different. It's going to bring in a little bit of biochemistry. Right? Not that we're going to talk how this actually occurs, but some of the molecules that a cell needs are two polar, meaning they have two wards of charge to go through the lipid membrane, or they're too large. So we would all agree that we need glucose in our cells. If you don't know this, glucose drives the production of ATP. That's one of the things it does. Right? Krebs cycle. I didn't want to say that word, but there it is. That's all I want to remember about the Krebs cycle. This stuff goes in, this goes out. What happens in the middle, just cover it up and act like, I already know that. It's like a rite of passage in some class. Learning the correct side. So some are too large or they're too polar. They can't get through the membrane. But we can still move them through in a passive process called facilitated diffusion. And for some of y'all first learning this, just because we're getting a little more detailed than we have to for my class on this, write passive up here, just so you remember, this is a passive process. And what happens is you have a molecule that's in the interstitial fluid or extracellular fluid, comes along, and it binds to a protein carrier. All right, what's another name? I'm trying to link everything this morning together. It's another name for a protein carrier on a membrane, an integral protein. So it's a type of carrier. And then it's going to be released onto the other side. So there's no energy used here, but in this case, we have, I'm going to call it glucose. So you have this molecule, and it comes along, and it has the ability, and they are specific for them, to bind with this receptor, or this integral protein, right? And so it's going to go in there, and this is the black box part of it. When that molecule interacts with that transport protein, it's going to undergo a conformational change, and if you notice it opened up on the inside now, and it will bring that molecule into the cell. So it kind of is open, the molecule interacts with it, and it goes through a conformational change, opens up and it comes in. It's called a carrier protein or a transport protein. Right? If you never, and I'll be with you, if you never understand what types of molecular bonds broke and moved to make all this happen without energy, you'll be fine the rest of your life. So quit going there. I know some of y'all are already picturing that because you like that stuff. But go talk to the biochemist about it if you really want to look at the molecular structures. All right. So those are all passive, and then we switch to active. So that's a great point for us to take a break, right? Then we'll get into our active transfer. Okay, break.
For like facilitated, so this one, there's no, use no energy. The liquid soluble has to fit.
Yes, yes.
In this case, I say this is it has to fit into this. Into this transfer. If this one wasn't, we try because there's different ones, like basically maybe a little bit of this membrane here.
So the easiest thing is, and the typo is this outside. The easiest thing to remember is at all point it wants to be so
Like plus, and then
outside
it's just all
higher concentration. You have negative water at the cell. Which has a higher concentration. Water versus cell has a higher concentration and outside has a lower concentration. Now to pull water from the outside of the cell it's all doing it. I think what using is that movement of sodium within the water. Diluting it just makes it which is why hypertonic solutions or ICP because you have like pulling all that
Now that makes more sense. We talked about it until we asked the question on the water, not the sodium. Thank you.
Sodium will always stay the same.
So we get you that for the first 2 weeks they have to be your bring the water producing. Which they would get.
there's all these concepts that you are. The fluid ice, solution ice, it will be a lot of
I'm still doing this again since late, but I'm only gonna do.
add an electrolyte in there to allow.
I think I'm gonna make my spread Tuesday, Wednesday, Thursday, and get into it leave Monday, Friday.
I fucked up though, because I wake up and I said, so the more you
I wake up and it's like 6:30. Yeah, no, I wake up and it's like 20 minutes. I have to like mess up my head.
I put my alarm and I'll put it over after it's just.
My little routine is like get myself go to the bathroom, go outside, pack my lunch, pack my laptop, go to coffee with her, bathroom, shave, get everything ready, my dog out, like 6:50-ish. Yeah. Yeah, it'll be fine. Oh, the, yeah. What, do you have a, do you plug that to your so the, you know how ones will have the 2 buttons in the middle? Yeah, so that's like the GFR, right? So like, the light will turn off from the GFR to what you have to figure out is where is that connected to? So like in my house, I had to call me because I said, "What's your bathroom?" And they're like, "Oh, the GFRI for all the bathrooms is in the store with the desk box. I found it down there. So yours, for your coffee, that outlet's probably make sure plug it into a different cord. There was yeah, no, so there's literally one in my kitchen, like the kitchen is like legit, like, you find that so that's great. I don't think it's that means it's a fuse and we'll say like the GFR. Sometimes they have like yeah, so you know, if you have to go this week through the breakers, that's how they shoot down. Which is why like I got tired of it. Yeah, like I freaking back to the guy like he fixed my head. I freaking the guy like he's like the technician for like stuff and he gets stuff on the wall and like the tours and stuff for the inspection and he gave us like a year worth I had an issue with my house in yeah, the whole shit, right? You guys literally said and then they said like an issue one is an AC issue. Like HVAC guy. Let's say it's not like the fire. We have gas back like this they're all like half this you have to well, yes, like I think like oh yeah, Ford will come to the like I got a memo, I got a little note right now. Yeah, that's fine. Okay. I never saw it. But listen, I made sure they I got the paper and said that's all
Alright, I was just commenting, it looks like we made it through our 10 slides, and that's awesome. And I wanna say this, just because I like y'all, is I love to talk about that stuff, but often, for some, especially as we go further on, you're not always listening to everything I say, we don't always have that interaction, as y'all go forward and get more on your minds, I get less questions. But when we start stop in an area like tonicity and have a good conversation about it, it's actually more, feels like you're learning more, right? We're discussing and we're talking. But then you like go home and you're studying and you remember that in your mind. I hear this feeling like, yeah, we talked about that a long time, so that must be like where all the questions are coming from. No. We had a discussion, and if y'all open up questions that are pertinent to it, I mean, if it was outside of kind of what we really need to know, I'll shut it down. But don't overemphasize the importance of tonicity, right? Just the base. It's good to understand it. I'm with you. I like to explain that, but we're really studying these transport mechanisms. And when I write questions, they come from the entirety. You know, if you want to pick sections, I used to have them divided up. You know, it was intro, then you have tissues, then you have genetics, and they're all separate. And when I wrote most of my tests, and I still do this, I pretty much take equally throughout. If there's an example where I'm going to pull from one section more than another, I will tell you, and that's only going to occur once. And that's going to be cranial nerves. Because if we don't learn anything else the whole time you're here, and you leave and you say, yeah, but I know cranial nerves, I'm fine with that. Those of you who practice medicine know what I mean. Cranial nerves are important. So if you don't ever understand tonicity, now that we talk about it so much, tonicities, if you were understanding all of that talk in basic sense, you'll really have a much better time when you get to emergency medicine. You get to start learning about how you would use those fluids and things effectively. Because this is not something the average medic, all that stuff we just talked about, the average medic should not be doing out in the battlefield, right? Depending on their training, I guess. All right. I'm talking about the medics that spend most of their time, like I did, out in the 25th Infantry Division, high-speed medic, lining up vehicles at the motor pool twice a day. Nobody moves the vehicles, why I gotta go back out and line them up? So, anyway. You know what that teaches you as a private? Quit asking questions you don't need to know. Just do it. You're doing nothing else. Just, I look back on it now and think about how irritated I would get. And now I'm like, I would do the same thing. Y'all just sitting around the office, go line the vehicles up again. Sergeant Major's coming out there. That was just a lie. Sergeant Major ain't going out at 4 o'clock on a Friday. Well, some of them are. But some will go out. Anyway, okay, no more digressing. Okay, active transport. We did our passives, and we got active transports. We're going to use energy. Okay. Somebody asked a question at the break, and we were describing these, and I can explain all of the active processes in one kind of statement. If we know that there are concentration gradients for every molecule of water, everything in the body has a concentration gradient, and the cell membranes establish that. And so we already kind of understand that. But there are examples and cases where we're going to need to move things against the concentration gradient. So take sodium. And we have sodium is higher outside a cell than it is inside. Right? So if we open up some type of channel in the membrane, sodium will be able to come in and diffuse down its concentration gradient passively, right? If it was open. But what if I had, I'm going to create big numbers because it helps you remember. I have 5,000 molecules of sodium outside the cell, whoops, and I have 10 molecules of sodium inside the cell. There's 5,000 outside, there's 10 inside. But for whatever purpose, just forget what purpose, I need to move, is this still working? I need to move sodium out of the cell. I want to take 3 of those 5 and move it outside the cell to where the 5,000 are. That's an active process. Active processes require energy to do that because we're moving against the concentration gradient. We're moving upriver now. Now we don't even need to do the slides because you understand it all. No, you got to know the kinds. Oh gosh. Who told me the belt thing? See, I put it back on my belt and I started doing it. He's right. Wait a minute. I wish I had known that a year ago. You're only as smart as you are educated. actually experienced. I don't know how to call it. Okay, so when you look at the polar charges and how many molecules, and we want to take them against their gradient, we can use primary active transport or we can use secondary active transport. You're going to hear a lot about it. Probably another class already has probably mentioned that. If not, almost every class this semester talks not only about primary active transport, they spend a lot of time on the most discussed integral protein out there is probably the sodium-potassium pump. So I already know that in biochem, you're going to discuss how many potassiums are moving out. You're going to look at ATP, ADP, movement. That's fine. They're going to do that. For here, I'm using this picture of the sodium-potassium pump just to give you an example of active transport, primary active transport. I don't need you to know at this point how many sodiums are moving and what ATP is doing. All I need you to know is that it requires ATP. Fair enough? Okay. So in order to do that, we're going to have a protein in here. In this case, we're calling it a sodium-potassium pump. And there are lots of different, there's calcium channel pumps, right? We're simplifying, but there are a lot of these within that membrane. And when you look at the fact that sodium and potassium are in different amounts inside and outside of the cell, this pump helps to establish that, along with other things. But it's going to help to maintain the difference of those ions inside and outside of the cell. So that's primary active transport. Bring sodium in, put potassium out. Right? We can send them both ways. That, and I keep going back. This requires the use of ATP, the body's energy. You must expend energy to do that. If you have no ATP present, it wouldn't happen. Right? In regards to a sodium-potassium pump. Now, when you look at secondary active transport, people try to confuse this. Okay? Because it's a little confusing when you say this, and we black box it a little bit, and secondary active transport kind of inadvertently, or not inadvertently, but it uses the energy of primary active transport. And the best way I know to explain that is if you take, hopefully I'll say this right. We have, let's do numbers. We have 10 sodium inside a cell. And we have 200 sodium outside of the cell. And so if we're going to take some of the 10 that are inside and pump it outside, we're going against its gradient. We're going to, that's primary active transport. We're going to use ATP to do that. So going back to what we first started talking about today, if I put more sodium outside the cell, have I increased the concentration gradient? Made it more steep? Yes, because there's more sodium outside, and sodium is very osmotic. And so I've increased that gradient. So if I've increased the gradient, and there are other ways than sodium-potassium pumps for sodium to get into a cell, I've increased the likelihood that if I open like a leakage channel for sodium, that more sodium is going to come in. So think of this, if I'm still tracking, if you're still tracking with me, if I pump sodium against its gradient, I'm building up more of kind of an osmotic pressure for sodium to go back in, right? The going back in is secondary active transport. So the energy for secondary active transport was established by moving sodium against its concentration gradient to make it higher outside the cell. And now we have more of a force to bring it back in. Yes, no, maybe. Okay. So once we know that, then you can just black box it and say, okay, well, primary active transport uses ATP, and that energy creates a difference in the balance of these ions, and that energy is kind of stored there so that we can use these secondary processes, which are called antiporters and symporters. So once you establish a little bit higher gradient outside of that cell, there are things, other integral proteins that are found in this plasma membrane that can take like sodium. It can open up and it's going to bring sodium back in. So now it's going to continue down the concentration gradient. But at the same time, that particular type of transport protein there with it can also move calcium out. In that case, if sodium goes in, calcium goes out of that same area, that's called an antiporter. They go against each other. Right? If it's a symporter on the other side here, a symporter is like sodium coming in back into the cell, and in this case, glucose decided to be a freeloader. Glucose will join sodium and come in at the same time. So glucose is sodium is sympathetic to glucose. Sees it sitting outside the cell, it's too large to get in, and says, tell you what, man, just get in the car with me. Right? I've already got the gas, we'll go in. Yeah.
So symporter, so if we have the secondary active transport, a symporter is basically one of those cations is just calling something else.
Exactly. Yeah. So sodium's the gassed up motorcycle coming back in. I don't know why I just thought this. And telling them, hey, glucose, jump on the back. Yeah, I don't know about y'all, but I'll ride on the back of the motorcycle. If I'm riding a motorcycle, I'll be driving. Right? Anyway, I don't ride motorcycles either, so I didn't mean to make it. I'm not this big motorcycle person. You know why I don't ride a motorcycle?
Yeah. Kitty transport?
No, I'm not worried about dying. But the way I die is not going to be on a motorcycle because y'all have been here long enough to see San Antonio. Everywhere you go in the world, you're going to complain about the other drivers because you're not used to it. But San Antonio stands unique. San Antonio there's people out there who do not have any insurance and they do not use blinkers. And we have a lot of exits, right? That's why I'm always complaining. Why don't you cut me off the exit? And you can exit 10 feet up. Anyway. But the problem with motorcycles, especially in San Antonio, is you can be the best motorcycle driver in the world, but if I'm in my Tahoe and they hit me, I'm going to be like, oh my gosh, I can't believe they hit the back quarter panel. That's all one piece. I can't replace that. Now I'm bondoing the crap out of this thing. If I'm on a motorcycle and they do that, I'm probably hurt pretty bad. That's my reason. I love driving a motorcycle and riding around. I ain't doing it because of that. Not for sure. It's a good carry. I like it. Alright. So the symporters, bring that in. I don't believe we got on the motorcycles, but I gotta stop that because so many people complain about my digressions. But I'll tell you this. It makes it a lot more fun. The problem is y'all like the digressions in the beginning, but as the iPad starts to beat you down, all you want to do is get through lectures so you can get home and study for hours, believe it or not. And you start to go, God, I wish Mr. Wright would just hammer through. And I will at sometimes. But I listen to seniors and juniors when they come back in the hallway and they talk to you. They tell you what you should, because by then I have no control over it. Once you leave my class, I don't control any grades, right? Barely control them here. And so people don't have to listen to you. They can say what they want to you now. So I listen to them when they go, hey, I didn't like it when you did this in class. Maybe you should change this. So I will always listen to that. Now, if one person says it, am I probably going to make a change? No. If I hear it every semester from a new group, when you talked about this, I didn't understand it, then I'm going to make that change. But other than that, most people tell me to keep the digressions up, but then the negative people put them on the critique all the time. You can see it's affecting me greatly. Transport and vesicles. We're still talking active transport processes. So the last part of that was using vesicles, and vesicles also must use ATP. Vesicular transport is a form of active transport. Circling that, I put a note there because you often forget that. So you're going to take a vesicle, and it forms, you're going to use the plasma membrane to make a vesicle, either to move things into the cell or outside of the cell. So you've heard of phagocytosis. where the cell membrane engulfs the bacteria, brings it in, that's budding off of the membrane and surrounding it. You can actually watch that in real time on like electron microscopes. It's kind of cool. But that's not the only type, because there's also receptor-mediated endocytosis. Endocytosis just means that we're going to take a molecule from outside the cell, move it inside the cell, using now a vesicle. And so it'll approach the cell, it'll move into the cell. Receptor-mediated endocytosis means that we're going to have a receptor that's looking for a particular molecule, and then it will do endocytosis. And then there's penocytosis, which is what's sometimes called bulk phase. Exocytosis is doing the opposite. Taking something that was made inside the cell, like a neurotransmitter, and moving it outside the cell. That will move to the wall, the cell membrane, and bud. The vesicle will fuse with the membrane and dump it into the exterior of the cell. And transcytosis is like in single layer areas like the lungs, the exchange of oxygen, carbon dioxide, different molecules. It can move through that single layer, so you'll do endocytosis, it'll move through the cell, and then do exocytosis without doing anything in the cell necessarily. Okay. So here's a picture of it. Well, you're welcome to make a note. I'm not asking you to diagram out this picture. It's definitely not going to be a study pic. Somebody remind me before I finish, so I'm not saying it here and disrupting this, to explain to you how I view using the study pics and the self-assessment that I posted. And I'll tell you how I set that up to be used. But I won't do it here. Okay, receptor-mediated endocytosis means that you have receptors on the outside of the cell that are looking for a specific particle, molecule, and when that molecule comes along, it's going to bind to that receptor. That's going to cause invagination of the membrane, and it will bring that molecule in because it interacted with the receptor. These are very specific. Once it brings it in there, the cell needs to use it, and so lysosomes break down molecules, or that molecule can be used like it is, but once you bring it in, it's going to break down what was brought in for use in the cell, using lysosomes. That's why I said that's all you need to know about the picture. I don't need you to label that thing. Phagocytosis, cell eating, means you're going to take the plasma membrane and wrap it around that bacteria until it engulfs it, and then it will bring it in, and once again, lysosomes are going to help you destroy and break that down. And we can use this for a lot of things. Worn-out cells, bacteria, viruses. The lysosomes are very powerful. Your lysosomes can even digest the entire cell they're in. Right? And then the last one of vesicular transport is called bulk phase endocytosis. I'm going to call this penocytosis because it'll help you to remember the difference between, it's a form of endocytosis. Well, there's several different forms. This just happens to be one of them. In this case, penocytosis refers to cell drinking. So the plasma membrane, for whatever reason, the reason is, I don't know a better example. You're the cell. You're an individual cell, and you decide, I'm thirsty. I don't have enough intracellular water content. For whatever reason, the cell is going to decide that. And when it does, it's going to take its membrane and just randomly take a big gulp of water. It's going to, just like you did phagocytosis, right? It's going to engulf a big portion, not big, but a portion of the interstitial fluid. And it will just send it out, and it'll grab all that water. So it wants to bring water in to increase intracellular water content. When it does that, is there anything else out there in that water? Molecules. Sodium, chloride, all those molecules are still there. And so when it takes a sip of the extracellular fluid and brings it in, it'll still bring in molecules. That's why it's called bulk phase endocytosis. It's bringing things in bulk. But what triggers it is you need to increase the cell water content. Exocytosis, we don't have to spend as much time on it, but it's your cells make things like neurotransmitters, they make proteins that are going to be used outside of the cell. If you need to get it outside of the cell, things that move around inside a cell are generally membrane bound in a vesicle. So you have a vesicle that's inside of the cell, it will move to the cell membrane, it will fuse with it, and then it will open up and dump its products to the exterior. That's exocytosis. And then transcytosis is just endocytosis and exocytosis combined. It goes into one side of the cell, goes out the other. That's it. Like I said, a lot of times, nothing. Normally, when you bring something into a cell, it's causing some type of, it's either being used by the cell or it's being interacted inside the cell. A lot of times with transcytosis, it just goes through the cell without really, it's still going to be membrane bound because when you do endocytosis and bring that in, you're going to have a vesicle now inside of the cell, right? And that vesicle is going to move, let's say, to the other side of the cell and dump it right back out into the exterior. It'll make more sense when you talk about like renal transport mechanisms and things. How you bring things across. If you haven't noticed yet, there are many different ways that molecules move. That's the confusing part. And that's what I'm trying to get home. All of that talk up to this point is just how we can move things. But we still have more to go on that. The rest of this right now, though, we're going to talk about the organelles that we find within the cell, or the cytoplasm of the cell. So we're switching gears here. So we said that cytoplasm, by definition, includes the cytosol, which is the water, and then the organelles. So now we're going to add to that and say, well, inside the cell, specifically inside where the cytosol is where all the chemical reactions are going to occur. And whatever energy is made or released by these actions. This is where we're going to see the building blocks for cell maintenance, structure, growth, which is another way of saying protein synthesis occurs inside the cells. Right? And then organelles are where we're going to spend a lot of time looking at the specific structures inside that cell. For the rest of the day and part of tomorrow, we'll still be on organelles. So if you look at an organelle such as the cytoskeleton, we need to define what do they do. The cytoskeleton helps determine the shape of a cell. It also provides for cellular movement. Like when you look at cells like white bacteria, white blood cells that can move through different areas. Right? They're going to use different types of the cytoskeleton for that occur, but it also organizes the inside of the cell, where certain things are. Right? It can move things around in the cell using the cytoskeleton. We're going to see a lot of cytoskeleton when we go through tissues, because when we look at different tissues, sometimes the cytoskeleton is used to anchor cells together. It might change into another type of thing, like in skin. When you look at skin, they call them tonofilaments, and they turn into keratin. Don't worry about it. You don't know what I'm talking about. Don't worry about that. But that we use for different things. So there's 3 different types. You have microfilaments, intermediate filaments, and then microtubules. Right? Microtubules will be used in genetics when we like pull things apart. But they're also used to transport things within a cell. Microtubules are like train tracks, or like the river walk, where the little boats are on the little track. Those through in on that, they're not just floating out there. So, if you go down to the river walk, I haven't been to the river walk in years. That's like riding a moped in Honolulu. You only do it once every night. Anybody who's been to Hawaii knows what I mean. Mopeds are crazy there. been a long time since I've been there, but I bet the mopeds are still there. And I bet people are still dying every day. Cytoskeleton. I left it as one slide here. In general, when we look at microfilaments, they're going to go along just inside of the cell membrane. If you look at intermediate filaments, they're kind of strewn throughout. And then if you look at microtubules, there's actually a microtubule organizing center. The microtubules will be in here creating all these tracks so we can move things around in the cell, to include chromosomes. Alright, so how many people know what, I'm just going to start out with it. What's this? I know it's an X, but what do we use that to represent when we talk about things that we're going? Chromosome. When I say it, you'll know chromosome. Right? That's the symbology for a chromosome. Actually, this is the symbology for a chromosome too. We're going to get into that tomorrow in genetics. But that X is a recognizable form that almost everybody that ever studied anatomy stuff goes, oh, when you put it in reference, what answer we're looking for? They go, yeah, that's what I picture a chromosome looks like. Well, that's exactly what happened when they looked at them under a microscope. I don't want to get into genetics, but during metaphase, when the chromosomes are lined up, they look like an X. There's a reason for that we'll get to. But the reason I'm bringing that up now is most of the time, when you look at a cell, the chromosomes are relaxed. And when they're relaxed, they're kind of uncoiled. In my mind, they look like cotton candy in the picture, right? Inside of that cell, we have a nucleus down in here. And then surround all of this out here is all of your chromosomes. But they don't look like an X right now because they're relaxed. They're laid out. They're sitting on a beanbag neck eating Cheetos. But they're relaxed. I use that one a lot. So it'll come back up. Ron White. Hilarious comedian. My favorite line of his ever. Anybody know Ron White? Yeah. I love the line. I didn't know how many people it was going to take to whoop my ass, but I knew how many they were going to use. I like that. That's a great statement. Okay. Nucleus. Another organelle found inside of there. Normally the largest organelle, usually, and underlined, there's only one. Not all cells only have one nucleus. It's not that common, but one fact we'll get to later is like skeletal muscle cells are multinucleated. Multinucleated cells will not go through mitosis. Right? So that means the skeletal muscle that you have right now, you were born with it. You're hitting the gym. We're going to get into that in muscles. When you hit the gym and you say, yeah, I'm building more muscle, you're not. You're enlarging the muscle cells you already had when you were born for skeletal muscle. I shouldn't even have said that. We're not going to get to that until next test. But for some of you are like, BS. We'll get there. We'll get to how that happens. I won't get that far ahead. There's a nuclear envelope surrounding. It's a double membrane sac that surrounds the nucleus. And in that membrane, you have nuclear pores. Nuclear pores are where things go into and out of the nucleus. Remember, the nucleus is sitting in the middle of the cell. Surrounding that nucleus is all of the cytosol, or cytoplasm as a whole, right? It's sitting in there. You need to exchange things in and out of that nucleus. It will go through these nuclear pores. Inside the nucleus, you have the nucleolus. Sounds similar, but nucleolus is a very specified structure that makes ribosomes. So you're going to make ribosomes in the nucleolus, which is in the nucleus. And then genes. The genes, of course, we know are found in the nucleus because that's the hereditary units. And then genes will be organized on chromosomes. Right? Chromosomes are long molecules of DNA, double-stranded molecules of DNA that we'll get into. So genetically we have chromosomes that have a bunch of genes on them, right? Traits, characteristics, instructions. I like genetics a little bit, not much, but I like as much as we're going to talk about. I'm not because I built the slides. So there's your nucleus, and inside that would be the nucleolus. We don't have a representation. Actually, we do have a representation. We do have a representation. I circled this earlier. This is the double membrane of the nucleus, and these are the pores. Then you're going to have that nucleolus in there. Earlier I called that the nucleus as an example, but it's not. That's the nucleolus. Alright. So what the nucleolus does specifically is going to produce both ribosomes. When you make ribosomes, you have a large ribosome and a small ribosome. They're called subunits. To have a functional ribosome, they must be together. But when they're made, they're separate. So the nucleolus produces both ribosomal subunits in the nucleolus. But they don't stay in there. People confuse this too. You do not store ribosomes in the nucleus or the nucleolus. They're made in the nucleolus. Think of it as they're immediately going to be transferred to the cytosol. Right? So that's the difference. You don't store ribosomes in the nucleolus. You make them and they move out to the cytosol. Because where you're going to find ribosomes is in the cytosol, because that's where we do protein synthesis. We don't do that in the nucleus. Right? Hopefully that makes sense. So ribosomes are where we're going to take amino acids and assemble them into proteins. That's called protein synthesis. Right? So most of the time, these are stored in the cytoplasm free. They're floating around, loose in the cytosol, the water of the cell, floating around out there. Sometimes they attach themselves to a membrane, specifically the rough endoplasmic reticulum. The endoplasmic reticulum, the rough is called, there's a smooth and a rough. The rough is called that because when they look at it, often there are ribosomes that have attached to it. But they can be attached to it and taken back off, right? I hope you're realizing I'm getting specific into ribosomes are made in the nucleolus, not stored. They're stored in the cytosol, but sometimes those that are stored in the cytosol are attached to the rough endoplasmic reticulum. So another way to picture that is you say, well, wait. For those linear thinkers, people are really thinking, well, wait. Every time I've looked at a microscope and looked at the endoplasmic reticulum, it's all, the rough endoplasmic reticulum is always rough. It never looks like the smooth. So you say, well then you should say they're equally stored on the endoplasmic reticulum, the rough endoplasmic reticulum, and in the cytosol. No. They're being inserted and taken back and forth from that endoplasmic reticulum. Most of them reside in the cytosol. Okay. So I use catchphrases a lot. So ribosomes are the sites of protein synthesis. I'm going to give you the 5-second version of protein synthesis. The biochem is going to give you like a 2-hour lecture. I don't know how long the lecture is on protein synthesis. But I do it here and in genetics to explain this. But for now, let's just get the parts. You have a large subunit and a small subunit. When they need to code a protein, or when they need to put amino acids in order, they will come together and form what's called a functional ribosome. They will join together. They're going to take amino acids, put them into a chain, and stack them up. Right? So they're going to reside in the cytosol or cytoplasm. In this case, you can use either word because it's out inside of that cell. They're going to reside out there in that cytoplasm until they're needed, separate. And then they'll come together when they're needed. So the small subunit is going to initiate translation. That will cause it to recruit the large subunit over to it. So they will join together. And in that case, the small subunit is going to read the mRNA, which is called messenger RNA, which is the instructions of how we should stack these amino acids. Now, I normally talk about this more in genetics, so I'd like to leave it for that, or further when we talk about protein synthesis. But for now, all I need you to get is that you have a large subunit, a small subunit. When a messenger RNA arrives with a message to build a protein, they will come together and they will build that protein. I'm going to explain more how that happens later. Right now, we're just listing organelles, so we don't want to get too far. Another organelle is the rough and smooth endoplasmic reticulum. It's smooth, it doesn't have ribosomes on it. If it's rough, it does. Okay? And so when we look at the rough endoplasmic reticulum that we find out here, it's normally attached to the nuclear envelope. Not always, but most of the time it is. You look at neurons, we'll see that's different. It's kind of these linear sacs, and then you have all these ribosomes that are attached to it. The smooth endoplasmic reticulum is more tubular. Right? So those are both types of endoplasmic reticulum, smooth and rough. And then we have the all-important Golgi complex. The Golgi complex is one of the first examples for us where we start to see that in anatomy, one of the difficult things when you're first learning anatomy is that so many things have different names. And so many things can be defined by their name. And so I try to, if you're ever confused, ask me which one I would use. But sometimes we need to know both names because people use it. It's like medicine. In medicine, we learn, like I mostly know a medicine if you'll tell me the generic name. Because I know what the ending is and I can associate it with a class. I may not know that it's called some brand name. You know, like people say, I never take Motrin. Really, what do you take for headaches? A lead. You just let them go. Just let them go. Okay? What you don't know won't hurt you. Anyway, yeah. Sorry, yeah. But the reason I said that is I like to give you examples. One of the hardest parts is the Golgi complex. You read different, go out on the internet, you'll see it called the Golgi body, Golgi complex, Golgi apparatus, Golgi. You can say Golgi too if you want. I told you yesterday I'm not an English person, but you can pronounce it however you'd like, as long as you can recognize it on test. For my view. So then people go, well, which one are you going to use? Well, I normally call it the Golgi complex. But I'm not going to split hairs. So in aspects like that, if you don't know when we're using interchangeable words, like which ones do you normally use? Because what you really want to know as a student is like, what am I looking for on the test, right? And here's what I always say. Like something like this, if it says Golgi, it's Golgi. There won't be Golgi choices between Golgi body, Golgi complex. They're all the same, right? So you just learn the main word there. Hopefully that helps you later. But if not, you need to ask me. Like, hey, when you know both names there, I'm fine with you asking me that. I'll tell you straight out which one's normally used or what I use on the test for that name. Because there are a lot of things named two different ways in anatomy. And I wasn't there. I know I'm getting older, but I wasn't there. I would have shut some of this down. We just stuck with one name. It's like the person in a meeting who brings up something that's not even on the agenda. It makes me stay there longer. We're not talking. I love colonels who throw people out of the room when they do that. That's not part of this meeting. Get the hell out. Okay, Golgi complex. When you look at this, its job is to receive immature proteins from the rough ER. Then you're like, well, wait, you said ribosomes make proteins. They do, but are ribosomes on the rough ER? Yeah. So proteins get made there. They're immature, so we need to mature them to use them. They're going to get packaged into vesicles, because vesicles are how things move inside of cells, and they're going to move from the rough ER to the Golgi complex. We're about to look at that. So this is called the packaging and export mechanism for the cell. The Golgi complex, I put on there, it can synthesize carbohydrates, but its job is to take immature proteins and mature them to use them. Right? And so what's going to happen is you're going to have the rough ER is going to make an immature protein. It's going to move to the Golgi complex. And it's going to fuse with that and dump its products into the Golgi complex. And we're going to black box a little bit of it. It's going to go through these linear things. And it's going to mature. The longer it stays in there, for whatever it might be, it matures into a mature protein. Once that occurs, it will create another vesicle. And it will bud off of that Golgi complex. Now there are names that we need to know. The side that the vesicle from the rough ER fuses with is called the cis phase, so the entry phase. And then the side that it leaves from is called the trans phase. Right? So an immature protein leaves the rough ER, arrives at the cis phase of the Golgi complex. It will go inside of the Golgi complex and be changed into a mature protein. It will leave or be transported off of the trans phase. Right? This is Dell computers. I don't know if it's still like this, so I'm going to keep saying this analogy until people tell me it's different. But I know in the beginning, Dell, they probably still don't, it's very prevalent now. When they first started, Dell's specialty was they would buy, or they still do, I'm sure, they'd buy computer parts from everybody, they put them together and put their own software on it, and then sell it. So the rough ER receives all sorts of different types of proteins. Well, so does Dell. Dell has a cis phase. It's the what do you call it? The receiving side of the warehouse. The receiving side of the warehouse at Dell is receiving, whatever, disk players, what do you call them, disk drives. They may be receiving optical drives, hard drives, all from different people, different things. And so they're not a mature computer yet. So Dell receives them in its cis phase of its receiving die. Goes inside. Inside of there, all of those get put together into a mature computer or a mature protein. Then it's going to leave or be transported out to the stores through the trans phase. Like the analogy, that's Dell. Wish I'd have thought of it. Right? I wouldn't be here. Nah, I'd probably still be here. Alright, the last part of this is, we're images of it. The vesicles that come off of the Golgi complex are either going to be secretory vesicles, meaning they're going to be released from the cell. So it'll leave the trans phase, move through a vesicle to the plasma membrane. It will fuse with that and be dumped outside of it, which is exocytosis, right? Another thing that can happen is that membrane doesn't exist forever. The plasma membrane, the phospholipid bilayer, those proteins, how did all that get out there? The cell made it. Right? Well, the cell needs to repair that. The cell needs to move. The cell has the ability to put, we've already discussed this today, and we understand that, I'm sure, like a sodium potassium pump is an integral protein. How do those integral proteins, I said they can be put there and they can be taken away by the cell, right? Where do they come from? This is where they come from. The cell makes them, puts them in a vesicle, and let's say it makes an integral protein that's a sodium potassium pump, and it wants to put it into the membrane. It'll move it in that vesicle, it will bud with the membrane, and it will just become part of the membrane. Right? That's how when I just generically say they're placed in it, that's how they move out there. That's called a membrane vesicle. It can also be more lipids and stuff for the membrane. And then transport vesicles are mature proteins and things that leave in a vesicle from the Golgi complex and go to other organelles inside of this cell. Because other organelles need to be maintained also. So those are the three ways. Alright, I'm going to speed up because I got to stop and point my I'm gonna do like 4 more slides, we'll get out of here, I'll get it out of here in time. So mitochondria, you're gonna spend a lot of time talking about mitochondria. Mitochondria are the powerhouse of the cell. Mitochondria are where cellular respiration occurs, that dreaded Krebs cycle. It's where we make ATP, body's energy. And so almost all of the ATP that's made in the body is made using mitochondria. And an interesting fact of them is they're self-replicating. So cells go through mitosis. Some cells go through mitosis. They can replicate themselves. And that's not so unique in the body. But what's unique about mitochondria is they contain their own DNA. So you can establish heredity using mitochondrial DNA. You've probably seen this if you watch as much crime shows as I do. And I've probably watched a lot out there. But you ever heard them say, they ran through Ancestry.com and they figured out that it has to be one of these two brothers. Because they only had a partial DNA match. But if they have enough, they can match maternal DNA from mitochondria. And that'll tell them enough to say, we know that whoever did this is a offspring of this mother. We can't tell which brother or sister or whatever did it. Well, you might be able to tell brother or sister, I don't know. You can't tell which sibling did it. So you gotta go find them all. It's another reason I don't put my DNA in Ancestry.com. I don't think I did anything, but there are a few years that I'm not sure about. So, I know, some of y'all, don't laugh at me, because some of y'all are with me. Some are like, yeah, I'm not doing it either. It doesn't matter, the military has you do. I cannot get off track here. I haven't talked here. Okay, I wanted to cover these things. I'm not going to do it. Forget it. I'm not going to cover these other stuff. That's the ending of Organelles, and I wanted to hit that point, but I don't want to take up y'all's time. So I can say, I love that people put their DNA in Ancestry.com. Anybody ever done that? They're coming to get you. Choose for crime and fit. Now I'm gonna finish with this. I had an E6 in Hawaii when I was a medic. I was like 84 at the time, and I was directed with the other medics to go and get blood samples for DNA for everybody in our unit. And if you ever looked in the back, I don't know if y'all still look at your paper medical charts, but back then, if you went to the back after you had this done, I forget what they call it, there's a little blotter with some blood drops on it. Any of you ever seen that in your chart? They used to have that. And they started doing this, that everybody had to have that, just back in the 90s, and I'm in Hawaii. And they said, okay, everybody's got to do this. We got to go draw blood, we got to drop them on there for every single person to get their DNA. Out of everybody we had in the unit, we had one E6 that said, I ain't doing it. Not giving you my DNA. And he went on for months. And legal was involved, and the command's involved. And he's really burning his career because he forgot one key factor about the military that sometimes sucks. And it's called UCMJ. Right? UCMJ is why it's good to be a civilian here. Because I'm not subject to UCMJ. Actually, I'm retired, so a little bit I am. Right? If you don't know that, wait until you retire. The point of this is, now I'm taking time. He fought that and fought that and fought that, and then they made him, they forced him, and legal got involved. He was to the point of being kicked out of the military. He finally said, okay, I'll do it. He was command directed. They brought him over with people. They had me do it. I pulled up his chart. I flipped to the back. He already had it in there. My goodness. You'd think he was 6 foot old. But, I don't know. That's a true story. Y'all have a good day.